I'm very excited to have this chance for an exchange of ideas about the intersection of conservation and academically based research. I greatly appreciate the interest in this topic shown by people working in plant conservation in Hawaii. I hope that all of you will find the topics we discuss to be interesting and of value to you in your work. As the title of my talk implies, I believe that collaboration across these two areas has the potential to benefit both conservation and basic science. Our work could not have been successful without the active collaboration of conservationists in Hawaii, and I hope that our work has benefited conservation as well. Although I have lived in California for many decades, I was born and raised in Michigan, and from a very early age was fascinated by plants. I feel at home in eastern deciduous forests, especially in the spring when ephemeral wildflowers put on their show. But the places I feel most connected to are the two areas where I've had the privilege of carrying out research. These locations, the Hawaiian Islands and the Sky Islands of the Sonoran Desert, despite their great differences, share features of island biogeography that make them wonderful places to study plant evolution. Both areas are threatened in many ways, and I hope that future generations will have the same chance to appreciate them as I have had. My first encounter with Schiedea in the field was at the end of the Waikamoi flume on Maui. Bob Hobby, shown on the right, had taken me there to see Claremontia because we thought at the time we were interested in their pollination biology. Claremontia was interesting, but I was amazed to see Schiedea diffusa hanging over the trail. You might wonder why I found this plant to be so impressive, especially if you compare flowers of Claremontia to those of Schiedea. Years before I met her, Ann Sakai, my long-term research collaborator, had entertained the idea of carrying out research on breeding system evolution in Schiedea after reading Sherwin Carlquist's discussion of breeding systems of this genus in Hawaii, A Natural History. Ann had collected some cuttings of Schiedea globosa from Makapu and still had them growing at her house, where I saw them years before seeing any Schiedea in the field. When I saw Schiedea diffusa in the field, I was utterly amazed to realize that something so different could be in the same genus, and Ann and I realized that Schiedea would provide a great system for investigating breeding system evolution. We also realized early on that we were not going to find very many Schiedea on our own. In fact, we have discovered exactly one population of Schiedea after hiking up the wrong ridge on West Maui and finding Schiedea salicaria at Pu'uhona. The way we were able to see all other Schiedea species was with the help of many field biologists on Oahu and other islands. The image here was taken after a memorable trip to Paholi Gulch with John Obata and Steve Perlman, where we saw Schiedea kaali. You can guess this image was taken a while back, and some of you may never have had the chance to meet John Obata. He knew every gulch in the Waianae Mountains and was extraordinarily generous in taking us to these areas. Field biologists throughout Hawaii were equally generous with their time. We made repeated trips to Haleakala Crater to find Schiedea haleakalensis in flower so we could determine the nature of the reproductive system. Art Medeiros was instrumental in helping us locate this species. We made several trips to Haleakala during the winter months when most species of Schiedea that occur in shrublands and on cliffs flower. Before considering the possibility that a subalpine species might be more likely to flower in the summer months, this proved to be correct when we realized that this species is dioecious once we found it. The first few years of field work in Hawaii were incredibly exciting for us because we were seeing species and sites for the first time and getting the most basic information about variation of breeding systems within Schiedea. As we accumulated information about Schiedea, we realized that variation in reproductive systems was even greater than we had guessed. Our initial goal was to study the evolution of separate sexes, and 10 species of Schiedea proved to have dioece or a related breeding system, and this allowed for comparative studies that provided insights into the evolution of dioece. For example, we were able to compare the ancestral condition of biotic pollination in hermaphroditic Schiedea litigati to wind pollination in Schiedea salicaria, where few females occur in populations, and nearly full dioece in Schiedea globosa, where most individuals in the population are either female or male plants. And you can see flowers of those individuals on the right. We also realized the presence of a second evolutionary trend from outcrossing biotically pollinated species to facultatively selfing species, shown in the upper right for Schiedea lichnoides, with substantial nectar production, to species with cleistogamous flowers, shown in the lower right, that are obligate selfers. Using these species, we were able to address the relationship of reproductive systems to the extent of inbreeding depression, an issue of fundamental importance in conservation of rare plants. From the outset, our work in Hawaii was encouraged by Warren Wagner, shown here with Ken Wood and Dave Lawrence. Warren had prepared the treatment of Schiedea for the Manual of Flowering Plants of Hawaii. As our understanding of reproductive systems of Schiedea increased, Warren suggested that it would be a good idea to monograph the genus. Understanding reproductive systems informed our understanding of the systematics of Schiedea, and our growing understanding of systematic relationships gave us new insights into reproductive systems. Ann and I never imagined we would spend decades working on Schiedea, but this genus has provided us with great opportunities to address interesting evolutionary questions and continues to surprise us. Shown here is a new species of Schiedea discovered by Tom Dement in the Laupahoehoe area of Hawaii Island. Tom and Josh Vandermark have been instrumental in obtaining and growing the material of this species that have allowed us, in collaboration with Warren Wagner, to determine that this is in fact an undescribed species, and also has provided the opportunity for Warren to write a species description. Although we were aware from the start that most Schiedea species were uncommon, and that some, such as Schiedea kaali and Schiedea haliacalensis, were extremely rare, we didn't think about actively participating in conservation efforts until we were working on Schiedea adamantis, which at the time was known from only a single population on Leahi. Our approach was to use the knowledge and materials that were outcomes of our research to promote conservation. We were certainly not the first to take this approach, and I used these images of a silver sword species and a cyanea to reference outstanding contributions to conservation of these lineages by Rob Robichaux and others to make this point. Our research on the quantitative genetic basis of reproductive traits in Schiedea required a large crossing program with as many representatives of the field population as possible to detect variation in these traits. We had representatives in the greenhouse grown from seeds collected in the field before 1996 and 1997, when drought resulted in tremendous mortality at Leahi, and the population size plummeted from over 200 individuals to fewer than 50 individuals. We reasoned that by using the excess seeds produced from the crosses shown in this diagram, we could provide genetically diverse propagules for restoration that would capture much of the original genetic variation in the population. Our first attempt at restoration, funded by the U.S. Fish and Wildlife Service, started in 1997. We had many partners, including Christine Willis, shown next to Ann on the rim of Leahi, and Dave Hopper, shown downslope, and also from Fish and Wildlife Service. The Division of Forestry and Wildlife helped by setting up an irrigation system, and Lion Arboretum stored the seeds from our original crossing program. We analyzed vegetation at the site where Schiedea adamantis occurred and picked areas on Leahi along the rim that were similar for restoration. We avoided the natural population for fear of damaging plants and used seeds because we couldn't easily outplant anything on the bare rock that predominated in this part of the crater. The irrigation system is shown here, along with metal tags where patches of seeds were planted. A few seedlings emerged, but in general, we could not provide enough water for germination or survival of the few seeds that germinated. In contrast, we also, at the same time, planted seeds in Cocoa Crater, where the Honolulu Botanic Garden set up an extensive irrigation system. These seeds germinated in large numbers, flowered rapidly, and many persisted for years. In 2012, a resurvey of the natural population on Leahi showed that only 19 individuals remained. One of the few large individuals is shown here. Teresa Coley, who worked with us on our quantitative genetic study of Schiedea adamantis, is comparing genetic variation before and after this severe decline in population size. Using the same seedlots that were stored at Lyon before this decline, another reintroduction effort was initiated with the support of PEP and the Division of Forestry and Wildlife. This effort was spearheaded by Susan Cheng Harbin, shown here with Talia Portner, and many other individuals participated as well. Greg Master from the Division of Forestry and Wildlife, shown here on the rim of Leahi with Anne, grew plants from seeds that were stored at Lyon and built an extensive irrigation system. Based on our previous experience, we chose an area on the lower slope of Leahi, below the natural population, where plants instead of seeds could be used for reestablishing the species, and where a water source was available to provide regular irrigation. With regular irrigation, plants established, they grew rapidly and produced flowers and seeds. Some of the plants reached large sizes, and because we saw recruitment of seedlings, we thought that we might finally have carried out a successful restoration effort. Unfortunately, a fire and loss of the irrigation system, combined with a lot of human traffic, as well as difficult access, eventually led to the failure of this second attempt as well. We all came to the conclusion that a site in the Ko'olau Mountains was the better option for restoring the species. We knew from molecular markers that some individuals of Schiedea globosa on windward Oahu contained DNA sequences of Schiedea adamantis, suggesting that Schiedea adamantis must have occurred in the Ko'olau Mountains, close to the current distribution of Schiedea globosa. An exciting discovery by Tim Kressick of a few Schiedea adamantis plants in one of the leeward gulches of the Ko'olau Mountains confirmed the wider distribution of this species and provides the potential for new genetic variation to incorporate in a third restoration attempt. Using genomic approaches, we will determine the extent of potential new variation from these plants. Tim's findings also suggest that the Ko'olau Mountains would be an appropriate site for another restoration effort. A third restoration effort, now underway in the Ko'olau Mountains at a higher, wetter site than Leahi, is supported by PEP and DOFA and is under the direction of Susan Cheng. Alex Loomis has used outplanted Schiedea adamantis for his conservation demographic studies, again using the same seeds from our initial quantitative genetics experiment. These plants are regenerating from seed, and it appears that this third attempt may have long-term success, suggesting that in restoration of rare species, persistence is essential, something that most of us have recognized for a long time. Our current study of Schiedea came about because of Lauren Weisenberger's work on the conservation genetics of Schiedea kaali. In addition to demonstrating strong heterosis and crosses between populations of the species, Lauren was the first to observe the regular influx of moths to flowers at dusk. These observations were obviously very exciting to us. Although we had inferred biotic pollination in many of the hermaphroditic species of Schiedea, we had not been able to make any observations of potential pollinators visiting flowers. We were so excited by these observations that we started thinking about the application of genomic approaches to phylogenetic reconstruction of relationships in Schiedea and the application of genomic approaches for addressing questions about gene flow and hybridization. With the help of many field biologists on Oahu, we were able to demonstrate that the moths visiting Schiedea kaali were all individuals of a new moth species, Pseudoschrankia brevipalpus. This tiny moth removes nectar from the small tubular nectaries unique to Schiedea flowers and transfers pollen in the process. Shortly after dusk, the population of Schiedea kaali was so flooded with moths that we described this time of night as moth madness. At some points in the evening, nearly every open Schiedea flower had a moth visit. It was truly a remarkable sight. We also found a striking correlation of Schiedea pollen grains and moth scales on stigmas of Schiedea kaali, providing us with an indirect method for assessing the likelihood of moth pollination in other species where we couldn't make field observations at night. Field observations by Lauren and her colleagues demonstrated that Pseudoschrankia brevipalpus also visits Schiedea hookeri. And because we found the same striking correlation of moth scales and Schiedea pollen grains on stigmas of Schiedea hookeri, we inferred that this species is also pollinated by Pseudoschrankia brevipalpus. We also found in choice tests in the field that Pseudoschrankia brevipalpus greatly prefers Schiedea kaali to wind-pollinated Schiedea species, as expected given differences in nectar production. These results indicate that pollen transfer from moth-pollinated to wind-pollinated species is unlikely in areas of sympatry for species with different modes of pollination. Our current research takes advantages of the powerful tools provided by genomics to gain a greater understanding of species relationships in Schiedea and help interpret breeding system evolution using an updated phylogenetic hypothesis. You can see in this most recent phylogeny using classic molecular markers that there simply isn't enough resolution to make these inferences. We also hope to extend information on native pollinators to other, presumably moth-pollinated Schiedea species, and then finally test hypotheses for the role of pollination biology in breeding system evolution. Here's the team involved in this current effort, which includes systematists, population biologists, experts in genomics, and conservation biologists. The team includes faculty, graduate students, and undergraduates at universities and colleges, research scientists at botanical gardens and museums, and those involved in implementing conservation efforts. Each of us is contributing in different ways, but ultimately, we need each other to gain a fuller understanding of the evolution of Schiedea. You will hear from other members of the team throughout this meeting. I would like to take this opportunity to describe a project that advances conservation efforts and our understanding of the importance of moth pollination throughout the genus. This goal is complicated by the extreme rarity of the Schiedea species most likely to be moth pollinated. Following the lead of Lauren's work on Schiedea kaali, we hope to create large populations of Schiedea kwaiensis that will attract native pollinators. We grew seeds from 5 founding individuals from the Mesic Forest on the west side of Kauai. These seeds were collected by Steve Perlman and Wendy Kishida. The plants that we grew had low fitness, almost certainly because they were produced by selfing of isolated single individuals. The plant on the left produced no seeds at all, while the plant on the right was reasonably fertile. Browses among progeny of different founders produced vigorous offspring that could be used for restoration. We had many partners in this project. For example, the few seeds that we could obtain from a single individual grown from the last surviving Nuololo Schiedea kwaiensis were sent to Lion Arboretum for germination and tissue culture. These plants were returned to us for incorporation into our crossing program. All of the seeds that we produced at UC Irvine were germinated at NTBG by Dustin Wolkos. Schiedea kwaiensis plants were grown to outplanting size at NTBG by Ashley Trask, and Steve Perlman helped in getting plants into the field. Schiedea kwaiensis plants were outplanted at the Pa'aiki exclosure on Kauai, as well as in Limahula Gardens. These plants are part of a demographic study by Alex Loomis, and we hope that they will attract native pollinators. We have not incorporated genotypes of Schiedea kwaiensis from Manoa Valley on the north side of Kauai because we are uncertain of the relationship of these plants to those from the west side of Kauai. Genomic analyses will provide further insights into the degree of similarity of plants from these different areas. The major goal of our current research is to determine the role of pollination biology and potential gene exchange between species. We will focus first on Schiedea legatii and Schiedea sarmentosa on Molokai because we know these species hybridize in the field, presumably have different pollination systems, and also differ in breeding systems. Schiedea sarmentosa on the left is a wind-pollinated species that has females in its populations, and we expect this species serves as a pollen donor to Schiedea legatii because of its wind pollination. In contrast, we believe that Schiedea legatii is moth-pollinated And based on our earlier choice tests, we predict that moths are not likely to visit Schiedea sarmentosa. Using field-collected seeds, we will determine the extent of gene flow using genomic markers, compare these results with evidence for past hybridization between these species. This approach has been used successfully to study hybridization in Schiedea. Eric Rolson, in collaboration with Warren Wagner and others, has used genomic approaches to verify hybridization between Schiedea species where morphological intermediacy and overlapping ranges had suggested this possibility. Our ultimate goal in Schiedea is to determine whether hybridization has led to the spread throughout the genus of male sterility alleles that cause the appearance of females in populations, or whether the independent evolution of separate sexes has occurred on multiple occasions. Thank you for listening. I hope that I've convinced you that academically based research on Hawaiian plants and conservation can work together to provide a greater understanding of how these plants have evolved and how we can use this knowledge to keep them from going extinct. I would like to thank the National Science Foundation for their support to the 4 institutions participating in this research. I also would like to take one last opportunity to thank the numerous individuals in Hawaii who have made this research possible. I can't show everyone who's helped us. If I tried, the screen would look like an enormous Zoom meeting. But thanks to all of you for your help and enthusiasm over the years.